So this would mean that I have tested these interactions. These are not identified interactions, but these are, let's say, one-to-all thing, right? And so on, right? So what we plot on the y-axis here is the 4C interaction frequency or 4C signal, 4C6 signal. And since this was our bait, right, the known region in 4C, remember we had this, known region, right, in all the loci. So we try to identify from this known reference region what are the unknown interacting partners. And this is your known region over here. And let us say we plotted the interaction frequency and it looked like that. Something like this. Okay. Now, what happens with the 4C data is that the interaction frequency decreases as a function of distance, right? And the reason behind this is the typical folding of the chromatin, which is the fractal organization. Okay, so the nearby loci will have very high tendency of interaction with the bait region, while as you go far away from the locus, the interaction frequency will decrease. Now that would mean that I cannot, if this interaction frequency is actually determined by, is a function of uh, genomic distance from the bait region. I call this as bait, right? This is my bait or reference point, you can call it. And as you see that the signals are actually decreasing as I go far away, but as I came very near to the bait, there are high frequency signals in the proximity. This implies that we cannot have one cutoff on the y-axis to say that this is my uh, cutoff to call a signal and rest all is noise. Okay. For example, in a typical chipstick experiment, what we do is the following thing. We have signals, noise, and like that, right? So what we do there is we simply take a cutoff. Not simply, there is statistics underlying it. We take a cutoff, which is just a straight line, horizontal line, okay, in parallel to the x-axis, and we say that anything above this is my peak signal, and rest all is my noise below this. I cannot have this. Why? Because of the very nature of the data that the interaction frequency at the proximal regions are high, no matter whether it's functional or not. Okay, and as we go away, it decreases, which means that we need to do something else. So what we do is the following. So we take this reference point, right? And we fit a lowest regression, which is the polynomial regression. Okay, so from as a function from this line, from this reference point, as a function of distance, we take the 4C signals. So what you could do is you can have distance here, genomic distance from D is distance from the reference point. Okay, for all the signals over here, and this is your 4C signal on y-axis. And of course, you're going to have a very high value near the reference point, which is here, right? Here is your bait. Okay, and then the signals are decreasing, right? So we need to fit a regression. Okay, so ideally we fit the regression on this data like that, and translate that regression coordinates onto this data. If you directly fit on this, there are other technical issues. So once we get this regression line, okay, which is polynomial regression from this line, we get something like that. It should be smooth line. Decreasing over time. Okay, now what we do is we calculate some kind of standard deviation from this regression line, generally the 3 standard deviation. Okay, so any signal which is crossing this 3 standard deviation will be called as peak or the signal like this one, and this one, let's see. Okay, they will be called as maybe this one also. Okay, they will be called as signal. So in this case, this is signal, and this is signal, right? Otherwise, we will end up having a lot of interactions just in nearby, which happens to be true with any locus. Okay, it will interact with the proximal region abundantly. So this is your, you can call it as a lowest regression, which is a polynomial regression thing. And this is 3 standard deviation from regression, from this regression line. Okay. Anything crossing this 3 standard deviation from the regression line is called as signal. So that's how you identify which region is interacting with your region of interest. So in this case, we have identified that this promoter and this promoter are interacting with my bait region, and there is some proximal region, maybe another secondary enhancer, which is also interacting with each other. This is the way we identify signals in the 4C data. Okay, so let's move on. So we learned about, we briefly discussed that there are, you know, there are techniques and there are corresponding scope of those techniques. 3C is one-to-one, 4C is one-to-all, and then we have many-to-many. This technique is, I'll, in the interest of time, today is the last class of this week, and I need to finish 2 things in this class. In the interest of time, I'm postponing the 5C thing. It's not that popular anymore. It used to be, it didn't make, it hasn't been very popular. 3C, 4C, and high C are the most popular one. When I see that we have some time left, okay, then I'll cover 5C also. If at all this is missed, then in experimental and computational techniques, I'll cover this again over there. 5C. So let's move on with all to all. So 1-to-1, 1-to-all, and then all to all. Okay, again, high C was given by Job Decker. So in between, if we talk about the authors here, this was Job Decker and his group. Basically, he did it single-handedly, and then whole industry of techniques followed. 4C was also took. And in Canada, there was another paper by Walter D. Land. From Netherlands. This fellow is in Sweden. The land job taker was in US. And high C is again job taker. While there were other techniques coming up, but I'm not talking about the popular most versions only. Okay, so I see. Okay, so we discussed 4C after the ligation step, right? So we did the ligation and we captured the surplus product. Okay, we need to go back to the digestion now. Okay, So let us say this is my, so you remember Hi-C has the scope to identify all-to-all interactions in an unbiased manner. So we have uh, these products out there, right? So these are digested products. You might wonder, what do I draw in blue color? These are protein complexes sitting out there. Okay, now before ligation, we do something else in 4C, sorry, in Hi-C. What we do is we add small adapters which are biotin
हाँ
related. Okay,
बहुत
so basically we are adding biotin on the open ends.
नहीं अभी बायोटिन लगा रहा है हाँ टेक्नो हाँ वो है तो टिपिकल पंजाबी ओवर हाँ बहुत ओवर स्मार्ट है गंदा ना
So biotin in green color we are
जूम नहीं आता इसे
having biotin.
उसके पास होगा खुद का झूमना नहीं है वो बहुत अच्छा पेपर ही छाप देगा
Okay, you can have small adapters with biotin labeling. And then you can do the ligation
तो
and
बोल रहा
followed
है
by
पूरा
the
टाइम
usual
देता
ligation thing.
है मतलब ये हर सेमेस्टर दो दो
Ligation in
कोर्सेज
dilution
लेता है
or
तो
in situ
इजी होता है
ligation.
तो इसको पेपर नहीं आता लाता वाह बहाना बड़ा अच्छा है वो तुझे कोर्स ही दो इसलिए दे तो तुमको कुछ काम
Yeah. So what will we get?
यार तो ये पेपर कब से लिखता है मतलब कब पेपर आए सिलेबस से नहीं आया लास्ट तो उसी का ही आया जब कैंसिल हाँ रांची का बैचमेट जो पेपर आया जिसका जल्दी पे पीएचडी हो गया था कैन नाम था ना कैन हाँ लोगों को सदमे लग गया
And our junction will have this biotin there, right? The reason we are adding biotin because the key to identify all-to-all interaction, we know neither of the interacting partner right now. We need to identify everything from ab initio, right? From scratch, we need to identify everything. So then it means the key thing in 3C, 4C has been the junction thing. So if we capture all the junctions, okay, we can identify what is there on the left hand side and the right hand side of the junction, upstream and downstream of the junction, and then we can map back those regions to the genome and find out what were those regions which came together and formed the junction in the Hi-C experiment. Okay, so you also may wonder that what happens to the other DNA, right? Why did we add biotin? We can identify junctions anyway, right? So we are using biotin so that we can pull down using antibody against it, right? Biotin streptodin interactions, and then we can pull it down. But the junction, why do we need to pull it down? Our solution will anyway will have junctions so that we can map the uh, upstream and downstream region of junction to the genome and find out to which places did they belong to. The idea is that we need to reduce the noise. For example, you may have these fragments out there, right, which are not bound with any protein complex or maybe bound with protein complex, but they're not capturing the proximity thing, right? And they will also get like biotinylated, right? These open ends. What happens is that since we are performing the ligation in dilution, right, in dilution, right? So what happens is that after we have done the ligation, these biotin moieties are getting stabilized because they got sandwiched between two DNA segments, right, after ligation. But there will be these fragments, open fragment out there again, which are biotinylated. But this biotin, which is just on the open ends, are actually very unstable. You can wash them away basically. If you now perform the washing step, with whatever buffer they use. So what happens is that we end up having only these complexes. And the other complexes, this DNA will not have the biotin. The biotin can be washed away. Okay, now you can pull down these fragments using antibody. Okay, we use the antibody against this and give me a minute. Sorry about the noise. This is the election time over here and people have gone crazy. Okay, so now we washed it and then we pull it down. We will only pull down the ones which are at the junction and is having stable biotin labeling. And once we pull it down, what we need to do is we need to chop it down further. Why do we need to chop it down? Because we don't need to, in the end, we will be using next generation sequencing and we want to reduce the sequencing cost, number one. And number two, we actually don't need so much of DNA to identify the interacting partner. So you use another restriction enzyme or DNA enzyme and further chop it down like that. So we will end up getting the junction and maybe 30, 40 or 50 base pair on either side of the junction so that it is suited to the next generation sequencing. So we have pulled down the fragments like this, which are smaller, right? And we had this biotin thing. This is your junction. And now what you do, you add your adapters, sequencing adapters. And then in your next generation sequencing, this will be first amplified and then sequenced next generation sequencing. To identify what is this region and what is this region, where do they map in the genome. Let's say they may map to distant region in the chromosome. One is mapping here, other is mapping here, and they were spatially together. That's why they got ligated. Proximity ligation, right? They were proximal, so they got ligated. And then you can find out that they were interacting. So they were like that. They were here, they were spatially proximal. Okay, now how do we represent this comprehensive data? Because this is all versus all, right? All the genomic regions versus all the genomic regions. So if you take one chromosome like this one, it is all the bins versus all the bins, basically all the restriction fragments versus all the restriction fragments. So generally what we do, we make a matrix. And this is self-to-self matrix, nothing else but a typical self-to-self matrix, right? Like you make a matrix of correlation values, covariance matrix. This is the same, let's say this is chromosome 1. Now you will have a certain resolution, maybe 500 resolution, 10 kb resolution, 20 kb, 30 kb, whatever. And you have those bins out there, right? Likewise, throughout the matrix. Right? And whatever the reads that you got from NGS, right? The read counts of each of these pairs, right? This is pair, right? Pair of this fragment versus this fragment. So read counts for each pair will be nothing but your interaction frequency, right? How often they come together in my cell sample, cell line sample, or whatever sample I had, right? So basically your diagonal would be very, very high because self-to-self interaction, or you can just suppress them that these are meaningless. But then you will have, let's say, if this region of this region over here is actually interacting with a region here of the same chromosome, right? Right, so then you're going to get a very high signal or value, interaction frequency value, read count. So read count can be represented as a heat map now. It's going to show this is interacting with this, right? This is interacting with this. So this versus this over here, there will be very high signal, right? And likewise, so this matrix will be symmetrical, isn't it? So over here and over here, very high signal, symmetrical. So this is the way we represent all interactions. Now each cell over here, each square in this grid represents interaction. For example, these two grids will be one interaction. There will be another one with this one and so on, right? So you can get a matrix for each chromosome, and then you can zoom in and make a matrix for your locus of interest of, let's say, 1 MB region, 2 MB region, and see where are your genes, where are your enhancer, whether they are interacting. Okay, and a typical way of representing Hi-C data is this matrix only. And you see on the left, there is one example. This is 1 MB region of mouse chromosome, and you see that each square here, there's small square over here, right? These squares are the interaction frequencies. So this is some 1 MB region versus same 1 MB region over here. What is peculiar here is that, of course, the diagonal has to be emptied because self-to-self is meaningless. But you see, along the diagonal, you have very high frequency of interactions, isn't it? Look at this square, right? You have these squares coming up along the diagonal. What does it tell you? What does it tell you? It tells you it's the same thing that we learned in the 4C. Where is 4C? This thing I said in, there is a very high interactions at the proximal regions, right? That's because of the very nature of chromatin folding. So this is one chromatin globule kind of thing. So this would be one chromatin globule, and then this is another one, this is another one, and this is another one, and then this is another one, bigger one, right? So that's what it is. So we call these complexes that we have of spatially proximal regions as topologically associated domains. Or tags. Now on the right hand side, you see another cartoon image just to illustrate that. Since I said that the matrix is symmetrical, the upper triangle of this matrix and the lower triangle represent the same information. So you actually don't need to represent as a square. What you can take, you can just take the upper triangle, right? You can just take this triangle, okay, and rotate it by 90 degree, and you get this thing, right? Just like the map that you have for linkage disequilibrium, right? You get this kind of maps out there. So the color in the heat map represents the interaction frequency, And those squares are now triangle there, right? The TADs are now like a triangle. And they have shown that, these triangle is one protein complex or TAD that we call, right? You see that this TAD has two sub-TADs also, like that. Okay, so that's one thing. The other thing is that these TADs contains genes which are generally, again somebody's typing. Yes, they remain same. Otherwise, why would I teach this? If you have something, if you have in a cell cycle, right? You already know that things get compressed during cell division, right? In the M phase, the chromosomes are the metaphase are the most condensed one, right? They are not in open chromatin state. So, and then they decompress it in G1 phase, right? And if the in the second round of cell cycle, okay, in the daughter cells, if the conformation of chromatin is not same as what it should be for the activity of right genes, then this is completely random thing. What you're learning in this course is the no. So biology is highly non-random. So non-random genome organization you are looking at. Okay, it has to. There is something memory, right? The memory of chromatin. How does the daughter cell know that these genes need to be silenced? Uh, I hope that was taught to you people. Was it taught to you how the chromatin states are inherited to daughter cells? No response. So, okay, so let me finish this thing and then we'll see if I can elaborate on that. So we have these complexes formed, right? Now they have identified different TADs using different algorithms. So how do those algorithms work? How do you identify TADs? The frequency of interactions of neighborhood, right? At some point that becomes your, right? For example, this bin over here versus this bin, the interaction frequency over here is low, right? So if this two neighboring bins have lower interaction frequency, they will be dropped. So if I'm calculating that interaction frequency of neighborhood windows, then if it is high, it will drop here. Sorry. So it will go somewhat like that, and wherever we have boundary, they will be dropped there, right? Drop there, and so on. So these becomes your TADs, TAD1, TAD2, TAD3. And likewise, you have more different algorithms. You also have network-based algorithm to identify TADs and so on. Okay, what is important to understand is that are these TADs functional? Why are they? Are they helping us in compartmentalizing the genomic functions? The answer is yes. And as every other phenomenon in biology, not absolute yes, but largely a big yes. What you see here are the different TADs again, right? So of course, the real picture is actually a little bit fuzzier here. You see that there's some kind of sub-TADs also. There could be one of this TAD, another TAD, and then you have sub-TADs and so on, right? Okay, so ignore this is TAD information, ignore this. So what you have over here is the following thing. You see S3K4 trimethylation, which is the transcribe regions, right? They are high on in this TAD. They are high on this TAD, which is this TAD, right? This TAD. Neighboring region has S3K27 trimethylation, which is high here. right, which is a separate TAD. So yes, it is segregating the distinct chromatin states into different domains. Can anybody guess why is that so? What is the advantage of this segregation?
So could you pardon the question?
What is the advantage? What is the physical significance of this kind of segregation that I just spoke about? You see some domain organization there, right? So there's one domain which is active, other domain is repressed now, and then in between you don't have any communication. That's the insulation that we call, or boundary elements. So if you look at the CTCF, it will be sitting over here, right? Sitting over here. Well, genomic functions, the genes which have similar functions or similar chromatin states, they are next to each other in the same tag. Well, the obvious reason that they are coming together in one domain is that their regulation is happening through concurrent opening of chromatin and concurrent closing of chromatin. It's a cost-effective way of doing so. If you have completely mixed up genes, where repressed or active genes can be anywhere in the genome and they're completely mixed up in a random manner, first of all, it will be a noisy process because everywhere the chromatin remodeling proteins have to open the chromatin for transcription and sporadically in the whole chromosome, right? And chromatin remodeling process itself is a noise prone. Okay, now in this case, they open chromatin remodeling complex, open the whole domain, right? And that state remains stable. So it reduces noise and it's a cost effective way of maintaining the chromatin states of similar genes. So developmental regulation would, you can now speculate how these domains are important. Once you go from one cell line to another cell line or one cell type to another cell type, you may say that, okay, these domain structure will change. Well, largely no. These domain structures are largely stable. Again, not absolutely, mostly stable from cell line to cell line, but their functions changes. What are those functions? These are their functions, right? This is active here, this is inactive here. Now, in another cell line, what could happen? This could swap the chromatin state. So let us say I have another cell line over here. And in that cell line, this is active. Okay, this is active. And this is repressed. So I will have K27 Me3 here and K36 Me3 here, right? So I'm not going to read the chat box. Okay, somebody keep on typing there despite my suggestion that you better speak up. Okay, it's very distracting. And then you ask a question which is simply not relevant to what I'm teaching right now. Okay, you can of course ask, maybe it's relevant 5 minutes ago, not now. Okay, so if you are so keen to ask question, please speak up. I'm not going to read the chat box. Okay, so this is another cell line and this was another cell line where we had K27 here and K36 here, right? So it's like these compartments are like a rooms, different rooms, classrooms, let's say, in isomerally, different classrooms. At the same time, in the parallel, two functions are going on, two classes are going on, right? And then it changes over time, right? Some other class happens in parallel into two rooms, right? So likewise here, in two different cell types, you can have different function of these domain, but the walls of your classroom doesn't break every time, right? You don't reconfigure or redesign your classroom structures. The architecture remains same. So by same analogy, these domains are rather stable, but their function, which is open chromatin versus closed chromatin, changes based on what is required for that cell type. Okay, so this is the way these domain function. And as I said, the boundaries are your boundary elements, and they are enriched with CTCF and cohesin protein, and I will come to that in a while. Okay, so this was high C. Now, what did we learn from all this? The first thing is, at very fundamental level, so I'm talking about 3C, 4C, and high C. Okay, what did we learn from all this? At very fundamental level, what we learn is the following thing. We have promoter and terminator. It turns out that at many genes, promoter interacts with the terminator. Okay, and so in 3D, how would it look like? We will have a loop, and these are called gene loops. And you will, of course, have protein complex sitting here. Right. So, can anybody suggest what is the physical significance of these gene loops? Why would promoter interact with the terminator? And this is particularly true for the smaller genes, like mostly the housekeeping genes and so on. I have already given you the hint.
To facilitate multiple rounds of transcription.
Yeah, so RNA pol2 would come, or some of you might say now you're going back to the textbook view of transcription. Other way around, this gene loop is actually mobile on RNA pol2. Okay, so when I say RNA pol2 is moving, it's actually the DNA is moving. Okay, so that's that model of RNA pol2 moving on DNA is still persisting. There are people who are working on that kind of model, or they are proponent of that model. But it seems the model where DNA is moving is seemingly correct. If you ask me, I would believe on this model, or I would be a proponent of the model where DNA is moving and RNA pol2 is stable. Okay, now this will move, right? And this will go and come back, and this will be recycled then, right? So this can be recycled, isn't it? So the pol2 will be recycling here. For what? For rapid transcription, right? And that's why I said that these genes are housekeeping genes more often with high transcription. So you want rapid transcription, and you have this gene loops where RNA pol2 are recycled, same one, until it goes really bad and the degradation is needed, right? So for the transcription of this gene, rapid transcription of this gene, these are your RNA copies. These gene loops have a physical significance out there. So that's at very fundamental level, the gene loops, okay? We call it as gene loops. My God, I don't remember the spelling of gene. Okay, and the other loop you already know, we are building up slowly. You have enhancer thing, right? So enhancer loops onto promoter. So I have a gene over here. Of course, they can coexist. The gene loop and the enhancer loops can coexist. And it turns out that it goes to a crazy level where you have multiple genes out there, and may have enhancers also in between. And their promoters are interacting with each other. And the enhancer is also interacting, and this enhancer may interact with multiple promoters also. Right? So they are making something called multi-gene complexes. Promoter to enhancer to promoter interaction is not new, right? EP loops, enhancer promoter loop. what was new, which came out of these assays that I just elaborated, promoter to promoter loops. Now, can you relate this to something which was taught to you in the last week? Why would one promoter interact with another one? Something like this. Well, it's simple. They are converging to a transcription factory, right? So there are multiple consequences of these. One consequence is that we have co-expression of these genes. The other is that, which is a bit thought-provoking, and that is, you remember there are promoters with enhancer functions, right? So enhancer like motors. So maybe one of these, maybe let's say this one is actually a enhancer like promoter, and it's actually regulating the T3. Okay, and how can you distinguish this? You can look at the chromatin states again and find out which are the enhancer-like promoters. And what you could do is you could test their potential. What you can do, you can have your luciferase gene assay, synthetic, and at the enhancer locus, you put your enhancer-like promoter, okay, and see if this is able to drive the transcription of your reporter gene. And it turns out, yes, in many cases, E promoter is driving the transcription of the target gene, reporter gene. But if you put a promoter-like promoter here, the promoter which is not having enhancer potential according to chromatin state, it doesn't regulate the target gene, the reporter gene. So these are the ways through which people try to validate that, yes, there are something called enhancer-like promoters. So what else did we learn? So okay, one thing, if we want to actually identify that are these interactions that I said that P1 is interacting with P2 and P2 is interacting with P3, are these really interacting at the transcription factory? It could be a repressive complex also, right? Is this a transcription factory? Is this RNA pol 2 loci? How would I know? Maybe this is actually a repressive complex, right? A heterochromatin complex. How do I know? What you could do is you can actually fish out from these complexes over here before adding biotinylation here. Or in this assay, you can actually filter out the complexes which are mediated by RNA polymerase 2 or your protein of interest. Your protein of interest could be any XYZ, could be a transcription factor, replication factor, CTCF, silencer binding protein, and so on, right? So what we do is we actually use CHIP. We use antibody against your factor of interest and filter out only the ones which are having your protein of interest at the complex, right? So now I know that my tube will only have this one. I have pulled down using antibody, and I will not take care of these kind of complexes which are mediated by some other thing. And if my antibody was against RNA pol 2, it is more likely that This is actually RNA pol 2 over here, right? Because I only filtered the complexes which were mediated by RNA pol. And then you have to validate it because this data is coming from uh, cell population, right? So we don't know if this is transcription factor is not determined at the cell population, it is determined at the single cell level. So then you have to actually do in situ experiment to show that yes, this is a Pol2 factory. You label RNA Pol2 phosphorylated form and you label your DNA elements with DNA fish. So combine immunofluorescence with DNA fish, you can actually find out whether your hypothesis is true or not. Right. So these kind of assays where you combine chip pair are known as high-C chip, high-C chromatin immunoprecipitation. There's another assay, similar assay called 3C. Sorry. There's another one which is more popular called Chia PET. Here, chromatin, CH stands for chromatin interaction analysis, IA. PET is a sequencing method, okay, which I'll not talk about in this class. So that this method necessarily mostly uses antibodies and pull out the complexes that they are interested in. Number one, and this is the only method which also uses sonication instead of restriction dilatation. So when I talk about 3C, 4C, and high C, that doesn't mean these are the only techniques. Okay, so these are not the only techniques. As I mentioned, Chia PET is another one, high C chip is another one. There is something called what is that? Yeah, capture high C, where let us say high C has all interactions, all to all interactions, but you're not interested in all to all interactions. What you're interested in is a subset of interactions. Let us say you have these are the fragments which got ligated, right? You're not interested in these, but only a few ones like this one and this one. Why? Because they were having promoter to promoter interactions, right? This was P1, P2, P3, P4. These were other interactions, enhancer to promoter or all other heterochromatin interaction that you were not interested. Then how can you pull out these? What you could do is you can use a chip again, like a microarray chip, where each spot is your different promoter sequence. Okay, and then you will be capturing these sequences which gets hybridized over these promoter elements, and only take the subpopulation now, which is captured one, which represents your sequences of interest, and follow up with the analysis. This would require, you might say, what's the use? You can analyze the whole genome thing and then filter out the regions which are promoter to promoter. Well, the benefit is that you need lesser sequencing here. Even if you do deep sequencing, you will be having very high confidence because your input sample was smaller, right? So lesser sequencing cost. For those reasons, people focus on capture high C kind of assay. And likewise, there are a number of different versions of high C and related assays. And I cannot talk about all the assays in this class. So this is what we learned so far about the, if you give me 5 minutes, let me elaborate a bit more, which will incorporate the whole hierarchy of findings so far. Okay. So for example, at the very small scale, we know that there are gene loops, right? And then we have enhancer loops, which can be accompanied by gene loop. And then we have promoter to promoter one, a bigger complex one, right? And then you have several genes and regulatory elements coming together and making a large CAD domain. CAD. And several of these TADs can come together in a phase separated complex. Okay. And this is another complex of several TADs. These are called compartments. Okay, so let me write down everything. Is gene loop at very basic level, EP loop, TAD, and these are called compartments. And finally, okay, let me tell you what is compartment. This is one phase separated complex of active chromatin or repressed chromatin, and this is another one of active chromatin. Okay, phase separation is involved in this process to make these compartments. Generally, these are called as A compartments. Active compartment is A, and inactive compartment is B, and they further fold into something called something called chromosomal territory. Okay, this is just a part of the nucleus. Chromosomal territories. You have different chromosomal territories. So the typical cruciform shape structure that you see in textbooks, okay, those are the metaphase chromosomes. Okay, so this kind of structure that you see now, or without these chromatids, you see this kind of structures, right? Even in the microscope, you can see that these are your metaphase chromosomes, right? Actually, in interphase, it remains dispersed like this. There is a defined chromosomal territory space for each chromosome, and the location of these territories are evolutionarily conserved. These are called chromosomal territories. Okay, so this is what the summary is so far. Okay, in the next class we will see somebody mentioned that are these the only methods of proximity ligation based for chromosome confirmation? No, we will briefly talk about yet another method in the next class, and then we will see a little bit on dynamics of this. Now, coming back to the question of what happens to this to the daughter cell? Well, they reconfigure into these territories and TAD domains back through the memory. For example, many of these things are not studied so far. For example, TADs disappear as you go into cell division, right? And they reappear in the early G1 phase. DNA replication is maintained to the daughter cells because hemimethylated states, one of the oldest strands during replication remains hemimethylated. And there are enzymes like DNA N-methyl transferase 2, which recognizes the hemimethylated DNA and methylates the newly synthesized DNA. That's how it is inherited for DNA methylation. It is not well studied for how chromatin, other histone modification, and others are maintained in cell division. Whether the new histones are recruited or old histones are re-recruited, there are studies which are supporting that the earlier histone, which was there in the parent DNA, is also getting recruited again to the daughter cells. Okay, and of course you need more histones because you are now dividing into multiple cells, 2 cells in each division. So you need more histones also. But the earlier ones are not simply degraded, but they are also being repeated again to the newly synthesized strands. And how does chromatin knows that in early G1 it has to go to this nuclear location, okay, and it has to be this conformation? There are a number of things there are something called licensing factors which license certain regions to be located in that region for those kind of functions. For example, the peripheral regions in the nucleus are the late replicating one, and then you have early replicating, which is in the center, the active regions. Replication, which origin of replication will be fired when is actually bookmarked. Okay, they are bookmarked in the early G1 phase. Okay, so although replication will happen in S phase, so there is something called bookmarking, okay, of regions which function as a memory. Same way, there are certain stone modifications which function as a bookmark for chromatin localization. So I will find out whether it was a histone modification or which histone modification function as a bookmarking thing in the next class. I forgot the name, or whether it was a transcription factor or some other factor. I am forgetting the precise name, but I will let you know. This field is very underexplored. The dynamics of chromatin reconfiguration during cell cycle has been worked out, and it is the same way as I said. It is open chromatin, and you see territorial organization, TAD organization in the G1 phase, in S1 phase also. And as you go in the G2 and the M phase, they get condensed and all TAD structure disappear, right? And then it reappears. The precise location, which locus should go where, is generally, it seems to be determined by some bookmarking factors. Okay, bookmarking things which doesn't change that drastically during cell cycle. Okay, even if it changes, it reestablish itself at the precise location very quickly. So there is something called bookmarking in the cell cycle. I'll mention for localization or chromatin localization, which histone modification is working as a bookmarking factor. I will let you know in the next class, or whether it's CTCF itself. I'm just missing that precise detail. I'll let you know in the next class. So that was all for the day. If I'm missing anything in this class, I will cover up in the next class. 5C is the carbon copy of 3C. I will upload the paper. It's not very difficult to understand. It's just that if I start talking about every technique, no matter how popular or unpopular a technique is, it's going to be not good for in interest of time. So we need to move on with the development also. The genomic imprinting and development I need to teach, and then Shravan will take over for diseases as well as DNA repair thing. So that was all for the day. If you have any question, please speak up.
Excuse me, sir.
Yeah.
Sir, I think regarding the chat, the reason why most of us use chat is because we don't want to disrupt the lecture while you are teaching,
No.
and we don't expect you to immediately clarify the doubts in the chat. We just want at least at the end of the lecture you see the chat.
No, so many of these are actually they are time-bound question. They are contextual question that you are asking about certain phenomena. I understand that, your thing, but I would personally like that you speak up, and it has been happening in earlier years. I don't mind that. So in fact, that will motivate your instructor also. If you, let's say I'm drawing something, it might be fine with people who are showing you the PowerPoint slides, but I am not very comfortable with PowerPoint slide because I don't have my freedom to draw the things in my way and explain the things on the go. So if I'm trying to focus on drawing something or to explain something, and all of a sudden things are popping up out there, and then when I go to question, it turns out that I taught that 5 minutes or 10 minutes ago, and then it's disrupting the flow. However, if you say, let's say, one thing is done. Let us say I am done with the how 4C is analyzed. Let's say I'm here. Okay, 4C is analyzed. And then when you see that I'm going to new slide and starting writing something, you can always ask something. I have a question about previous thing, and like you can find a time window where you see that I'm not speaking or I'm moving to the next part. Then you can ask the question. Even in the end, let's say you want to ask now, I would appreciate if you speak up.
So then, can I, if you don't mind, so my question was in the figure that you showed, there was uh, there was a tad with only one edge of the triangle darker and the other edge was not as dark. Not this tad, the other one. Yeah, here. So how can we visualize a tad like this because it's
One.
not uniform?
Which one?
So here in the middle triangle and in the yeah the other edge of the triangle is not dark. Only one edge is like dark.
I think you're talking about
The larger tad.
this one, right?
Yes, sir.
Yeah. So this is like a streak. Like there is one streak going on. It seems like the element which was located over here, right, is kind of going over the whole locus over here, right? So this locus is interacting with this locus over here, and this locus is moving over. It's like an enhancer promoter interaction. Okay. Did you get this? If you have
Kind of.
only
Yes,
one
sir.
streak out there, it means that one of the element is just moving over other, right? These are
Okay,
generally,
sir.
and this is like a tracking thing. It's interacting with, it tells you that it's interacting with this region also, and then this region also, and then this region also, right? That's what it's telling. So it can be like, I can give you where does this happen. Generally, this if you have a gene loop thing, right? So you will have interaction of this region with this region and this region and this region and this region and this region. Why? Because all two is actually traversing over this, right? In a loop form, right? So that's where you get these streaks. Generally, these are enhancer promoter interactions with the, it's like a tracking thing. Something is tracking. Maybe it's searching for a high affinity one and stopping where it's finding high affinity one like this one. Sometimes you have a isolated dot. I don't know whether we have here. For example, sometimes we get a structure like this. We have like this. See, and you just have, right now you saw a streak over here, right? Sometimes you will just find one thing here and very intense thing here. It tells you just this region is just interacting with this region with one loop, right? That's what it means.
Sir, and to translate this into the figure, like how you drew the loop, wouldn't a square matrix be helpful rather than just one like the?
Yeah, this is right now is visualization. In the next class, I will tell you that we don't stop just about these observation that I talk about the TATs and the multi-gene complexes and all that. And this is the final conclusion that I have. You can actually make a 3D genome model. Let me show you the picture in the next class. I will tell you how do we make it. Just give me 1 minute and I'm done.
पागल है क्या?
Okay, look at this. This is the 3D model of human genome. Okay, each color represents the uh, chromosome, one chromosomal territory. And this is modeled from HiC data. And in the next class, I'll tell you how do we get this from HiC data. Now they have just separated each chromosome for visualization purpose. All right, so that was it for the day, and I'll tell you in the next class from this matrix that I said, this matrix, how do we get 3D model? It's not very difficult to, the class, the method will not be computationally intensive. I will just tell you the concept. And it's even a layman can appreciate that, yeah, this is the way it can be modeled. There can be multiple methods to model it. I'll tell you one easy one. Okay, is there any more question? So this topic is so vast these days. Okay, the field has progressed very rapidly, and there are many things which can be taught. In fact, I can put an independent course on three-dimensional chromatin organization. But I will just talk about the fundamental thing in this course, which changes the the way we think about genome. Okay, if there's no more question, I'm stopping the class.
Recording stopped.